Talk will be two parts. The first one will be about like basic biology information about chromosomes, cell divisions, and cytogenetics. And then after that, we can talk about the implications of this information and understanding the chromosomal abnormalities in Down syndrome. So first, most of us know that our cells, our somatic cells, contain 46 chromosomes. They are arranged in 23 pairs. The pairs from 1 to 22, we call them the autosomes, and these are identical in males and females. And then we have the XX and XY depending on the gender. And the pair of each chromosome, we call them homologous chromosomes or homologs. They are identical under microscope, and they contain similar genes. So the chromosomes is composed of DNA and protein. So the double helix DNA will complex with different types of proteins, and this will form a chromatin. And normally this, like not during cell division at any given time, these chromatin are distributed homogeneously all over the nucleus. And they only condense to form chromosomes during cell division. So only we can visualize the chromosomes only during cell division. Otherwise, these chromatin are distributed all over the nucleus. And we have two types of cell division, the regular, what we call it mitosis, and the meiosis. In mitosis, we have the original cell will divide into two daughter cells that are identical. The amount of DNA will initially be replicated and then will be distributed evenly. So the daughter cells will have the same amount of DNA and number of chromosomes as the original cell. And the division goes through prophase, metaphase, anaphase, and telophase, and we will end with having two daughter cells. This type of cell division is called, as we said, mitosis, and it happens during cell growth and differentiation. But the other type that happens only during the formation of sperms and ova in germline cells, where we have two consecutive division, the meiosis 1 and meiosis 2. During the first one, the number of chromosomes are reduced by half. So the original cell we have it diploid, and after first division we have the haploid cells. The second division is exactly as mitosis, where we have distribution of the chromosomes among daughter cells. So we end having these 4 daughter cells that have only 23 chromosomes instead of 24 chromosomes. In spermatogenesis, or the formation of sperms, we start with the primary spermatocyte, and after the first meiosis, we end with 2 secondary spermatocytes. Each one will contain only 23 chromosomes. And then after that, we have the second meiosis ending in secondary spermatocytes, which will mature into mature sperms. And this process starts after sexual maturity and continues most of the life in males. However, oops, did I do something wrong? Yeah. Sorry. However, the story is little different for oogenesis. The process starts during the prenatal life. So during prenatal life, the primary oocyte starts the first meiosis. However, it stops at prophase. So that when a female is born, all her cells are stopped during the prophase, and it will remains arrested in this phase for many years till the time of ovulation. When ovulation takes place, the first meiosis will continue, and we will get the secondary oocyte, and then meiosis 2 will start but will not finish unless the egg is fertilized. So the important part here that the cell is arrested in prophase 1 for many years. Some cells will be arrested for 20, 30, 40 years till the time of ovulation, and this one has very important clinical implication. Then we'll talk a little about cytogenetics, which is the study of simply chromosomes, the structure of a chromosome, structural abnormalities of chromosomes, how to look at the chromosomes. And if we have any chromosomal abnormalities, if we have abnormalities in the chromosomes, then we have chromosomal diseases or chromosomal abnormalities like Down syndrome. And chromosomal abnormalities can result in distinctive syndromes and miscarriages, and also it's implicated in congenital malformation, some malignancies also. And if we need to test the chromosomes, we do chromosomal analysis or karyotyping. And as we said, we cannot see the chromosomes unless the cell is dividing. So we take a blood sample, we separate white blood cells because these cells can divide. We put them in culture media, stimulate them to grow, and then at one point stimulate them to divide. And then at one point we add something to stop the cell during metaphase, which is the time where these chromosomes are really very visible in a nice way. Then these cells are placed in hypotonic media, so it will burst, and chromosomes will go outside, and then they can be fixed, stains, and a picture like this is taken. In the past, the cytogenetists used to cut by hand these different chromosomes and align them to every two similar chromosomes beside each other. Nowadays, there are like Photoshop programs that can do that, and then chromosomes are arranged from chromosome 1, the longer, to chromosome 22, the smaller, plus the XX or XY chromosomes. And so this is the karyotype, which is, this one is normal. And with staining, the chromosomes, now chromosomes are different by their length, by the banding, white and dark band, and by the presence of the centromere. And if you see here, we have like 5 chromosomes, which are 13, 14, 15, 21, and 22. We call them acrocentric because the centromere is located at the end of the chromosome. So all the other chromosomes, they have short arm and long arm, but here the short arm is very small. And again, this one has another clinical implication that we will come to in a minute. Okay, so Down syndrome was clinically described in 1866, but it took like about 100 years to understand the molecular mechanism, or that Down syndrome is linked to chromosomal abnormalities. And this was the initial, this was like what typically we see in Down syndrome, 3 copies of a chromosome 21. This can be easily be seen by regular karyotyping. Now, although Down syndrome, as we will hear from the coming talks, it's a clinical diagnosis, still we need to do karyotype for confirmation, and very importantly to know the type of a chromosomal abnormality in Down syndrome that we will discuss now, because this will help in counseling and assessing the recurrent risk. Though really sometimes the type is not important in determining the severity of the disease. So about the types of chromosomal abnormalities in Down syndrome, we have 3 major types, either trisomy 21 or Robertsonian translocation or mosaic. And there is a small percentage for partial trisomy 21, and I'll spend the coming 10 or 15 minutes talking about these different types. So in trisomy 21, which is the most common that we see in about like 94 or 95% of the cases, if you do karyotype, all what you see is 3 copies of chromosome 21. And in this type of trisomy, trisomy 21 is like the overall incidence of Down syndrome is something between 1 in 600 to 800. But if you look by the age, the incidence starts to increase dramatically between the age of 30 and 35. And it may reach like up to 4% around the age of 45. So the incidence of a trisomy 21 increases with increased maternal age. And in fact, this fact is due to the etiology of a trisomy 21. The trisomy 21 most of the time, or 90% of the time, occurs due what we call it non-disjunction in the during the formation of oocyte. And if you remember, we said that the oocyte is frozen in this stage of prophase 1. Now, if ovulation took place at the age of 20 years, then that means this cell has been in this stage for 20 years. So there is a chance that the separation of the chromosomes didn't go in the right way, and 2 of chromosome 21 will go to 1 ova. On one of the cells. Now, this chance will be higher if the ovulation took place at the age of 30 years or 40 years. So with increasing time, the chance of having a non-disjunction or having an oocyte with 2 chromosome 21 will be higher. And of course, with a third chromosome 21 coming from the sperm, we will have 3 copies of chromosome 21. There is a small percentage where the sperm actually has 2 copies of chromosome 21, and it's like less than 10% where we can have a trisomy 21 due to abnormalities in the sperm. Now, if a lady has a child with a trisomy 21, the recurrent risk is very small. This is why it's important really to document that. So if she is less than 30 or 35, the risk is something between 1 to 2%. However, if the lady is older than 35, then the risk is similar to her regular risk, because as we said, the risk of having trisomy 21 is higher after 35. So the risk doesn't really increase significantly. It's the same because it's already high. Now, like around 3% of children with Down syndrome, if you do chromosomal analysis for them and you just count the chromosomes, they have like normal number of chromosomes. They have 46 chromosomes. However, still they have 3 copies of chromosome 21. The third copy is attached to another chromosome. And if you remember, we said we have some chromosomes, we call them acrocentric, where they only have the long chromosome. So typically what happens, and chromosome 21 is acrocentric, so chromosome 21 can be attached to other acrocentric chromosome. Chromosome 14 or 20 may be the common, but it can be attached to any other acrocentric chromosome. So if you count, then yes, they are 46, but one of these 46 chromosomes actually are 2 chromosomes attached to each other. A chromosome 14, for example, with a 21. So you have 3 copies of a chromosome 21. And this can happen de novo, like the parents have normal karyotype, but also the important part that the mother or the father can have what we call it balanced translocation. And I'll show you pictures for this. And in this situation, the recurrent risk is much higher than what you would expect from the regular trisomy 21. So I think these figures, so this is a mother. If you count here the carrier of balanced translocation, if you count the chromosomes, they are 45 chromosomes instead of 46, because one of the chromosomes is like this one, 14 and 21 are attached to each other. So this person has 2 copies of chromosome 14, 2 copies of chromosome 21. So this person has the right amount of genetic material. This is why we call it balanced. There is no extra chromosome in this person. It's the problem that 14 and 21 are attached. So this person doesn't have any complication. So if this lady has this balanced translocation, then the problem is during the formation of the oocytes, because there are 6 possibilities of the oocyte to be formed. And so if 14 and 21 went to 1 ova, then this will give us like normal oocyte, and this will result in normal offspring. While if the translocated chromosome goes to 1 cell, so again, this will be normal, but the carrier will have a balanced translocation, so she will be like her mother. But this is the problem. If we get this translocated chromosome in addition to chromosome 21. So if this forms an ova and then a sperm with chromosome 21 came here, fertilized this egg, then we will have 3 copies of chromosome 21. Now, the other 3 possibilities will not get any viable offspring. So theoretically, you may think that the risk of having children with Down syndrome here is 33%, but actually there is like advantages for the other 2 cells in survival. So the risk is only 10 to 15% for females. Now, so if we have a female with balanced translocation, then 10 to 15% risk for her children to have Down syndrome, which is much higher than just the regular trisomy 21. Now, for males, the risk even is lower, maybe like something around 3 to 5%. So if a male carries balanced translocation, the risk of having children with Down syndrome is small, around 3%. And this is what we see in karyotype. So this is a male with 2 copies of chromosome 14, 2 copies of chromosome 21, normal male. And here a female, phenotypically normal. She doesn't have any complaints, the mother, because she has, this is 14 and 21 and 21. So she has all the chromosomes, but the problem here is 21 and 14 are attached to each other. Now, the offspring will get 14 and 21 from the father, and if the offspring took this translocated chromosome and 21, then like by count, if you count them, they are 46 chromosomes, but there is an extra chromosome 21 here attached to chromosome 14. So we have 214, 221, and this is the third 21. So the child will have classic picture of trisomy 21, or Down syndrome due to Robertsonian translocation. Now, this is very rare that we may have 2 chromosomes of 21 attached to each other. I mean, this can, if the mother really carries these, or the mother or father carries this translocation, 21-21 translocation, then the recurrent risk will be 100%, because the ova will either carry the two copies of chromosome 21 or null, no copies of 21. And if you don't have 21, then it's not compatible with life. It will not survive the fertilized egg. But in this situation, if the mother or the father are carrier for 21 translocation, then all their offspring will be affected with Down syndrome. But sometimes this translocation happens de novo in the affected or in the child with trisomy 21. Finally, there is a small percentage of children with Down syndrome who have mosaic trisomy 21. That means not all their cells will have 3 copies of chromosome 21. Some of the cells will have 3 copies, other cells will have only two copies. So typically, these children will have milder phenotype, and the phenotype is really variable. It depends on when the abnormality takes place. So here we have like a normal sperm, normal egg, we have normal fertilized egg, and then the cell starts to divide. And at one point during cell division, we have non-disjunction, and 3 copies of chromosome 21 went to one cell, while 1 copy went to another cell. The 1 copy, again, these are not viable, so this cell will not divide, but this cell will continue dividing. Now, so we will have mixture of cells that have trisomy 21 and cells that have the normal 2 copies of chromosome 2, and it depends how early this abnormality takes place, if all cell lines will be affected, or just single cell line will be affected, or only limited cells will be affected. Finally, in very, very rare situation, you see a child and with features of Down syndrome, but when you do karyotype, you count the chromosomes, they are 46, you look at translocations, you don't have a translocation. So in these situations, sometimes if you look more carefully, it's what we call it partial trisomy 21. One of the chromosome, now we have 2 copies of chromosome 21, one of them, a small piece of a chromosome 21 can be duplicated. So if one piece is duplicated, then, and there is the same piece is present on the other chromosome, then you'll have 3 copies of this part of a chromosome, not the whole chromosome 21, but only part of a chromosome 21 will have 3 copies of that part. And this can be picked by karyotyping, because you'll find one of the 21 chromosome has extra piece. Sometimes the duplication is too small, and this is even more rare, and you will have completely normal karyotype. But if you do other sophisticated methodology like chromosome microarray, you'll find duplication, small duplication that cannot be seen, smaller than the ability to be seen by a regular karyotype, and that means you have 3 copies of small tiny piece of chromosome 21 that gives you features similar to the feature that we see in trisomy 21. So this will be I think this will be my last slide, and these are the references, and thank you for your attention.